My name is Sumanta Goswami. I'm a professor at Einstein College of Medicine. I have joint appointments between medical college as well as the university. So I first of all want to thank Sat for everything that he's doing. I'm a great fan of his. I mean, I wish I had something like this when I was growing up. I got into research way later. So now that you're exposing young minds to research earlier, I'm sure it's going to be a fruit. So I've had the pleasure of hosting 2 young women last year. And this year also I'm hosting 2 young women. Both of them are here. I'll try to talk a little bit about what they're doing. But please bear in mind, the research that I do takes many, many years of training. So students coming and staying with me for 2 months, at best, get a slight flavor of what research is like. The data that they generate usually goes into optimizing some method for a student who is going to do future research. So as far as productivity is concerned, I do not focus a lot on that when the students come to my lab. What I try to focus on is try to make them understand what breast cancer research is about, why we are doing it, how we are approaching it, what are the different problems that we are facing, and why is it after at least 50 years of research, we still have ladies dying of breast cancer. So most of the students who work with me come back with a deep appreciation of the problems of breast cancer research and how we are approaching it, rather than exactly getting some data. Of course, there are exceptions. I've had students who work just one summer with me, but he's a part of a publication, but those are more exceptions rather than rule. So usually, in order to get a publication from my laboratory, students have to work much more than just a summer. Okay, so I'll talk about breast cancer metastasis and drug resistance. So briefly, it's needless to say why cancer research in general, and breast cancer in particular, is important. All of us have lost some family member, some friend, our mothers, sisters, daughters, if you're unfortunate, in breast cancer. So fortunately, the good news is that in the last couple of years, the total number of patients dying out of breast cancer is reducing. That's a very good news. Why? Because over the last about 30 years or so, number of research has provided excellent drugs, early detection for those who have access to healthcare. This is very important to note. The numbers that I've given up there includes both people who have access to healthcare as well as those who do not have access to healthcare. So early detection in breast cancer, as you all know, is the key. So, what I'd like to impose on you today is that the absolute number of patients have gone down, but what has increased dramatically in the last 10 years or so are the number of patients dying out of metastatic cancer as well as drug-resistant cancer. Metastatic cancer is when cancer spreads. Okay, all of you know that cancer is a disease where you have suddenly uncontrolled growth. I don't know whether that phrase is true or not. That phrase is true or not anymore, but that's pretty much basically what it is. So today, what is happening is in the field of breast cancer, if we detect it early enough, and the lady has access to healthcare, or the man for that matter, do you guys know that at least about 1,800 men die of breast cancer every year? So if you didn't, then next time, if you ever hear a man with breast cancer, don't laugh. It's a reality. It's a reality. About 1,800 men die out of breast cancer every year in the United States alone. So, if the lady has access to healthcare and it's detected early enough, then the chances are that you would be treated surgery, chemotherapy, radiotherapy. But the problem is, if the cancer has spread to other parts of the body, then it's very difficult to cure, because the cancer has then acquired some unique features which make them more difficult to treat. The research in my laboratory is actually looking at these 2 unique features. What makes a cancer cell spread? What makes it drug resistant? Okay? I'll try to tell you in about 40 minutes from now on what exactly what they do. Can we have the lights a little down here? Because I'm going to show some movies and fluorescence images. So if anybody can control the lights a little down, it would be great. Thank you. Okay. So metastasis is a process when cancer spreads from the primary tumor into the local milieu and invades into the blood vessel, travels a long distance, survives the blood, travels a long distance, and comes out of blood vessel or lymphatic vessel and forms a distant metastasis. Okay? So as you can see, I already described that it's a multi-step process. Okay? So the approach that has been taken to handle cancer are anti-proliferative drugs. What I mean by anti-proliferative drugs are drugs that prevent the cells from proliferating. Another approach that was taken recently are protease inhibitors. Protease inhibitors prevent the cancer from spreading because you know when a cell has to move into the local milieu, it has to break the protein that is around. So if you can inhibit the spread, you can sort of keep the cancer under control. Another set of drugs that are in use right now are anti-metastatic drugs, which prevent the intravasation, as we call it, when a cell has to move into the blood vessel. Now, as you can see, each of these steps are essential for cancer to metastasize. Our laboratory is looking at a whole gambit of approaches by which we can prevent cancer from spreading. Now, since this is a sort of an educational program, I decided to put in this slide, which by a step-by-step tells you how cancer progresses and how it spreads. So the first step that happens is hyperproliferation. The number of cells increase, uncontrolled cell growth. The next step that happens is, as the tumor grows, as the solid tumor grows, the inner core of it becomes deprived of food, oxygen, and everything. So because of this, what can I say, hypoxic region in the middle, and oxidative stress occurs. The oxidative stress, because it's lack of oxygen, lack of food, increases angiogenesis. What I mean by increased angiogenesis is new blood vessels are formed. The exact same procedure happens in the embryonic stage of our life. When we were in our mother's wombs, a limb had to form. When a limb had to form, it's a small part first. The limb will slowly grow forward. So new blood vessels were forming. Cancer utilizes exactly the same, not exactly, but very much similar mechanisms by which it is able to generate new blood vessels so that those blood vessels will provide oxygen to these hypoxic regions. When that happens, the newly forming blood vessels are leaky blood vessels. They allow the cancer cell to enter the blood vessel and thereby increase the blood burden. Finally, the cells will move out of the blood vessel and form a cancer. Okay? So the approach that I'm going to talk about today is going to look at only the invasion and intravasation area. There's a huge group of scientists who are working on each of these steps that I've outlined here. My laboratory is only focused on invasion and intravasation. So in order to study these, you will need either cell models or animal models, or preferably both. So, because you cannot do any experiment on human beings. You have to develop a model in animals or in the laboratory with cell cultures, and once you work on those, only after you have established a drug or a technique which works in all these cases can you go in for a human trial. Okay? So in our laboratory, we have two types of models which we use. One of them is a cell model, another is an animal model.